Hello friends, welcome to our online platform of ABC of Food. This is lecture number 2 and in this we will be studying about preservation of food by heating. Heat transfer is one of the most important unit operations in the food industry. It is an energy transfer phenomena. As we know, heat is the form of energy that is transferred between two substances at different temperatures. The direction of energy flow is from the substance of higher temperature to the substance of a lower temperature. Nearly every process requires either heat input or heat removal to alter the physical, chemical, and storage characteristics of the product. For example, in storage of fruits, vegetables, and dairy products, heat is removed to decrease the temperature and to increase the storage life of the product. Whereas in other case, heat is given to change the characteristics of a product. For example, by cooking, which gives texture, color, and flavor to the food. Principles of heat transfer. There are three types of heat transfer. Firstly, via solid material, known as conduction. Secondly, via liquid and gases, known as convection. And thirdly, via electromagnetical waves, known as radiation method. In conduction, heat transfer occurs when thermal energy is transferred from one molecule to the other molecule. In convection, the molecules are free to move about, resulting in mixing of warmer and cooler portions of the same material. In radiation, it occurs by means of electromagnetic radiations emitted from one body and absorbed by another body. Thermal processing. It is defined as the combination of time and temperature required to eliminate a desired number of microorganisms from a food product. The word thermal itself refers to a process involving heat, and heating food is an effective way of preserving. The main objectives are: it destroys the microbial activity, it reduces enzymes activity, and third, very important, it improves the shelf life of a product. Apart from these, there are some undesirable changes also, like degradation of nutrients, flavor, and texture. Now we will be studying about factors which affect the heat resistance of a microorganisms. Firstly, the age or growth of a cell. The stage of growth of a vegetative cells affects their heat resistance. Secondly, the initial concentration of spores or cells. More the number of spores and cells present, greater is the heat resistance and hence more time is required. Third, the medium in which growth has occurred. It plays an important role in production of highly resistant spores. If the spores have grown in nutrient rich medium, then high temperature will be required for killing of spores. Fourth is pH. Spores are least resistant in acidic medium, followed by alkali medium and maximum neutral medium. Fifth is sugar and salt. Sugar in higher concentration protects the spores from heat and therefore requires more high temperature and longer time for destruction of spores. Where salt solutions, it decreases the resistance, therefore less time is required. Now we will study about the types of thermal processing. Firstly, cooking. The primary objective is to produce more palatable food. Second, blanching. It is a pre-thermal treatment given before drying or freezing to inactivate the enzymes. Third is pasteurization. It is done with the aim to kill the pathogenic microorganisms. Fourth is sterilization. It is done with the aim of complete destruction of microorganisms including their spores. Cooking. It is a common preservation technique since cooked foods generally can be stored for a longer period than uncooked foods. Refrigerated storage followed by cooking is a common household method of preservation. The primary objective is to produce more palatable food. It includes the cooking, baking, roasting, boiling, and frying. Blanching. It is a mild heat treatment given to vegetables and fruits before drying or freezing and then cooled rapidly to near ambient temperatures for their inactivation of oxidative enzymes such as catalase, peroxidase, polyphenol oxidase. It also cleans food materials, decreases the microbial load, and loosens the tissue. There are mainly two types of method which is commonly used is water blanching and steam blanching. In water blanching, in this fruits and vegetables soaked in boiling water for 1 to 5 minutes depending on the product. And in steam blanching, in this the food material is passed through a tunnel full of steam using conveyor belts. Other types of method which is not commonly used is microwave blanching, vacuum blanching, and third is individual quick blanching. Pasteurization. It is a process in which foods are heated to a temperature below the boiling point of water for a definite period, that is below 100 degrees centigrade. It destroys the pathogenic organisms associated with food and extends the shelf life of product by decreasing the microbial population and inactivating some enzymes. The choice of type and temperature is dependent on the type of food. For example, for milk is 63 centigrade for 30 minutes. For beer is 65 to 68 degree centigrade for 20 minutes. It is not a sterile process. It means that they can still contain vegetative spores and organisms which are still capable for growth. Sterilization. It is a controlled thermal treatment in which foods are heated at a sufficiently high temperature. Temperature is above 100 degrees centigrade, usually ranging from 110 to 121 degrees centigrade, depending on the type of food and size of a container. It completely eliminates or destroys all the microorganisms, including thermoresistant spores in the food. Now we will see the difference between the blanching, pasteurization, and sterilization. In blanching, it is a mild heat treatment given to food before drying or freezing to inactivate the natural food enzymes and also to destroy some microorganisms. Pasteurization, it is a relatively mild heat treatment in which food is heated below 100 degrees centigrade. It is used to minimize possible health hazards from pathogenic microorganisms and to extend the shelf life of food for several days. In sterilization, the foods are heated at a high temperature and for a long time to destroy microbial and enzymes activity. As a result, sterilized foods have a shelf life in excess of 6 months at ambient temperatures. So thanks for watching, and if you like this video, please share and subscribe my channel.